out of time at the end, and I don't want to forget to acknowledge all my terrific collaborators. I just want to call out some of these folks. The carbon cycling work is very interdisciplinary. So all of these projects, there are a number of collaborators. So the first portion on the North Atlantic blooms, working with my former postdoc, Cheng-Feng Le, and Xuanwen Hu, who's at University of South Florida. The second piece is mainly on the intermediate trophic levels, is mainly collaboration with Brad Penta at the Naval Research Lab and Adam Greer, who's now at the University of Georgia. And then the work in the Gulf, there's a whole host of folks there. Hanji Wang is a postdoc, and then Weijin Tsai, University of Delaware, Steve Lorenz, Kanchan Mady, and Brian Roberts. And then for the last piece, I'll just briefly talk about a project that is recently underway that involves a host of folks from our institution, Ronnie Baker, Brian Swankowski, and Sean Powers, as well as folks at Mississippi State, at Auburn University, and at North Carolina State. So this is kind of my scientific sandbox, looking at the ocean carbon cycle. There's a lot of different conceptual models out there that I could have used, but this is one that I've developed during my career, really. And we've put this together not only to illustrate what we're talking about when we're talking about the carbon cycle, but also because this has now been developed into a numerical model. So mathematical equations that represent all of these state variables and the processes that link them. So just quickly, I'm going to run through this because we're going to talk about some of these interactions today. So the first part of the talk, when we talk about the North Atlantic blooms, we're really going to be talking about the coupling and the role that the atmosphere and winds play on mixing the water column. And in the open ocean in the North Atlantic, the mixing of that water column provides nutrients, and it also creates a change in the layer of the stratification, which can then induce the spring bloom. The second part of the talk is going to focus here sort of in this center box on the zooplankton. And so a real weakness of our understanding of the carbon cycle from my perspective is how these intermediate trophic levels both participate as sort of the groups that are eating all of this material that's being generated by primary producers, but then also how they control the carbon cycle from the top down. And then when we transition to the Gulf work, I'll be talking a bit about sediment contributions to carbon cycling. And then hopefully with the last part that we're now doing in Mobile Bay, it's a bit more of a synthesis and bringing all of this together. So we can really take that complicated figure and distill it down to something much more simple that I think most everybody should be able to get their arms around. Basically what I'm interested in is how external forcing, and that forcing can be climate, it can be human stressors, it can be river discharge, etc. How that forcing affects these important state variables, nutrient concentrations, phytoplankton biomass and community, zooplankton biomass and community, and then the organic matter, the detritus that's created from that. And in turn, how do these affect oxygen and CO2 dynamics? Oxygen is something that I'm interested in because of hypoxia, which we'll talk about when we talk about the Gulf work. And over the last decade or so, I've been tying in carbon dioxide and acidification as it's related in these processes. So we can take that conceptual model that I showed before that was very complicated, that included all the compartments, including the sediments, and we can adopt or adapt that for particular problems. And so the North Atlantic spring bloom is something that I've been working on for a few years now, as I mentioned with my former postdoc, Cheng-Feng Le. And really in that work, what we're after there is trying to link atmospheric phenomena to changing wind patterns. In turn, how do those winds affect the depth of the mixed layer and the deepening and shallowing of that mixed layer, the introduction of nutrients, which are then believed in turn to control the phytoplankton bloom and ultimately the biological pump in the ocean. So just to kind of orient ourselves where we're talking about, this is a map of the North Atlantic from about 20 degrees latitude up to the pole, the North Pole. And the reason that we're interested in this spring bloom is because it is the largest bloom event on the planet. The North Atlantic spring bloom accounts for approximately 20% of the global ocean carbon dioxide uptake. We've been doing some work in a couple of study regions. Today I'm going to focus on this sort of subtropical region here located between 30 degrees north and 40 degrees north in the central Atlantic. But we're also looking at some work on eastern boundary current over here off the coast of Africa in the Canary upwelling system. I'll save that for another time. I think the first part today is really illustrative of what we're trying to do here. Okay, so just a little bit of a primer on the North Atlantic phytoplankton bloom. So there's several hypotheses now about what triggers and ultimately controls the bloom. The oldest of those is the critical depth hypothesis. You've probably learned about this in biological oceanography. It's attributed to Sverdrup, who was a physical oceanographer. But basically the idea here is that the bloom is limited by the mixing depth. So when we have mixing during the wintertime, the phytoplankton are diluted because the mixing depth increases. And then they're also being mixed below the depth where light is available. So they're no longer able to photosynthesize. And then the trigger for the spring bloom is as that mixed layer depth begins to shallow and it reaches a critical depth where the light is penetrating all the way to the bottom of the mixed layer, the bloom is triggered. So that's sort of the classical paradigm for the bloom. And then more recently, there's been some other hypotheses, a little bit more complicated. I'm not going to go into those today. There's a lot of very strong opinions and positions about these hypotheses. But what we wanted to do was to back up and to look at this initiation of the bloom. All of these hypotheses begin with this mixed layer deepening aspect. So this is the crucial trigger, physical trigger for the bloom. Biologically, what happens after that, I think is still open for debate. But we all of these hypotheses agree that the mixed layer deepening is the important first step. So can we figure out what is driving that mixed layer deepening in the subtropical Atlantic? So what I'm going to propose today, and this is work, we have a couple of manuscripts that are currently in review. It's not published yet. But what we've proposed is that there's a teleconnection to the Arctic. And when I say teleconnection, that means that there's something happening at a spatial location that is very distant from where the bloom is occurring that is driving the variability in the northern Atlantic. And what we have focused on is this phenomena called Greenland blocking. Okay, so Greenland blocking is an atmospheric phenomena, and it's been proposed as the mechanism that regulates zonal wind variability in the high Arctic, high latitudes. And what we'll demonstrate is that it actually affects the lower latitudes and drives the timing and the duration of the spring bloom. So I'm just going to, since we're not atmospheric scientists, I'm just going to quickly show you a couple of the states for this Greenland blocking phenomena. So this is a site called earthnullschool.net. You've probably used it or seen it in your classes, but here's Greenland up here, okay? And so under this scenario, there's a high pressure system that's occupying, basically sitting over Greenland. And based on the rotation of this high pressure system, there's a large portion of the jet stream, which is coming here across North America, that is actually being shunted up into the Arctic. And so this is a concern for warming and melting in the Arctic. But for the purposes of today's talk, this is actually during this setup, when we have this sort of high pressure system over Greenland, the winds are northern. So if we look at where our box was, it was centered on 30 degrees to 40 degrees. So down sort of in this lower center part of the map. So let's look at a different scenario, a different Greenland blocking. Here, in this scenario, we have a low pressure system that's now sitting over Greenland. And what we can see happens is when we have this low pressure system, it diverts the jet stream southward. And now we see this very strong zonal wind pattern throughout our study area in the North Atlantic. And so we wanted to see if we could link that to phytoplankton blooms based on the idea that those stronger winds may be creating a deeper mixed layer. My students are probably sick of hearing this, but I say it all the time. We don't need to collect new data to do a lot of studies. There's so much data that's out there these days. And I really like this study because we didn't have any ship time to go out in the North Atlantic. We didn't collect any of our own data, but I think we're able to put together a really novel and compelling story based on available data sources. So the data that we used were meteorological data that were retrieved from the National Center for Environmental Prediction, their reanalysis products. Two of those products that we used are the geopotential height at 500 hectopascal. If you're not an atmospheric scientist, you probably don't know what a hectopascal is. It's equivalent to a millibar. So basically we're looking at height in the atmosphere at about 5 kilometers, and the variation in that height, as you'll see, will be one of the primary factors that we'll be looking at. We also used the data from NCEP on winds. We used all of the ocean color data that were available from CWIFS and MODIS. And with these ocean color data, we were able to generate synoptic Atlantic Ocean-wide phytoplankton time series. We used the mixed layer depth product that's created out of Oregon State. We utilized data from the Bermuda Atlantic Time Series Station, which is located in our study region at about 35 or 36 degrees north, maybe even a little bit higher, 37, 38. Nonetheless, it's a long-term time series of data that's been collected by the Bermuda Biological Institution, and it was useful here for validating our analysis. And with these data, we were able to explore and put together a series of correlations, really, that connect Greenland blocking to zonal winds, to mixed layer depth, and ultimately to phytoplankton biomass. All right, so a little bit more about this Greenland blocking. So there's an index that's calculated that describes this Greenland blocking phenomenon. It's called the Greenland Blocking Index. And it's calculated as the height of the 500 hectopascal level. So you can imagine that if there's a high pressure system sitting at a location, that that higher pressure is going to drive that 500 hectopascal level down. So it's going to decrease that level. And then when we have low pressure, the air is going to expand and it'll make that level go up. So really what this index is an index of high pressure or low pressure dominating over Greenland. The blocking index is calculated over this grid from 80 degrees north to 60 degrees north, and then from 20 degrees west to 80 degrees, I'm sorry, from 20 degrees west to 80 degrees west. So we calculated over the period of record from the 1990s to the present, we calculated this Greenland blocking index based on these data. All right, so here's a little bit of what the data actually looked like. So here I'm showing the anomaly. So the index itself varies, the height of that 500 hectopascal layer varies between about 4.8 kilometers and 5.2 kilometers. So it's more, well, it makes it easier to visualize by looking at the anomaly of that. So here's our area up here in blue where we calculated this blocking index. We can see the bright red indicate, in this case, that the Greenland blocking is high. And so when the index is high, that actually means there's a low pressure system there. And under this low pressure system, we can see these zonal wind patterns extend well down into the lower latitudes, even to 20 degrees north or so, so south of where we are, right? So Mobile's at 30 degrees north. This blocking pattern drives the jet stream south and creates higher winds over our study region. So this is an example of an extreme blocking index. If we look at a more average condition, we can see that under these medium conditions, we're still driving the jet stream southward and creating these zonal winds across our study area. And then when we look, when this anomaly, when this index is low, we actually see a wind reversal. Now the winds are blowing to the west and they're a lot smaller. So let's look and see what the phytoplankton biomass, the response to these different Greenland blocking patterns. In the first figure here, the red indicates that the phytoplankton in the spring bloom is higher than average. So the dark red is 50% higher than average. The dark blue is 50% lower than average. So we can see under this high Greenland blocking anomaly, we see an extreme phytoplankton bloom, spring bloom in the North Atlantic. And then in a year when we have a low GBI, we can see that there's actually very low phytoplankton, small phytoplankton bloom in those conditions. So how are these related? Okay, so here's the correlation of the winter GBI and the spring chlorophyll A anomaly. So the darker red shows higher correlation. So we can see that there's a strong correlation between that Greenland blocking and the bloom. And we see it not only in the satellite data, which are shown here, as well as here's the average chlorophyll A averaged over our study area. And then here's the data from the BATS time series. And so there's a very strong correlation between this Greenland blocking index and the chlorophyll A. So let's see if we can break that down a bit and figure out what's going on. How are these phenomena related mechanistically? This is just another view to look at it in time series mode. So again, now we're calculating the bloom duration and the bloom intensity. These are just two metrics of the strength of the bloom. But if we start like out here in the more recent past, we see that there's a strong coherence in these time series. It's interesting though, when we get back into the mid and early 2000s, it seems to break down a bit. And we just got clobbered by a reviewer in a recent review that we're responding to now, who really took us to task because we didn't explain what was going on in this period. We didn't really, frankly, know. So we've looked at it in some more detail. And it turns out that some other climatologists have noted that the Greenland blocking was not behaving as typical during those years. And that was associated with a period of rapid ice melt and now a transition to open water in some areas around Greenland. So we're still trying to figure out how that ties into our story. But I think we have a better understanding now of why this pattern isn't in synchrony all the time, although from over the last decade and a half or so, there's a very nice coherence in the signals. All right, so this is showing now the correlation with the wind speed. So the wind in the east, from the east to the west direction. So we're starting to mechanistically break this down a bit. So the Greenland blocking index strongly related to the wind fields. And I'm not going to show you the winds related to the vertical mixing. We've put together this kind of conceptual model now of what we think is happening that starts with the Greenland blocking. So under the conditions where we have this strong low pressure system, it diverts the jet stream southward, equatorward. That creates higher zonal winds over our study area. Higher winds equate to greater vertical mixing, mixing depth. That increases nutrient supply, and in turn, ultimately then, when the light is available in the spring, we see a biomass bloom. So that's some exciting work. I think that'll be pretty impactful when it's published. It's a little bit different than some of the other stories that are out there. Most of the previous work has been done at higher latitudes in the North Atlantic, between about 40 degrees north and 60 degrees north. And sort of the climate change story in that area of the Atlantic is that it's thought that the bloom may decrease in the future because sea surface warming is going to increase stratification strength, and the mixed layer deepening is not going to occur. So our mechanism a little bit further to the south is a different story than what is for the farther North Atlantic. All right, so now I'm going to transition to the study of intermediate trophic levels also in the North Atlantic. where we're really focusing now in this sort of where I would say the highest area of uncertainty is in our understanding of the carbon cycle is the interaction with intermediate trophic levels and how those can both drive things from top down and be controlled from bottom up. So this is a video that sort of started it all. My collaborator and friend Brad Penta at the Naval Research Lab collected this data from just a GoPro camera from some work that he was doing, and he came back and he showed it to me, and we got really excited because you'll see that there's just this tremendous patch of biomass. In this case, it's all jellyfish. But you know, at the beginning, they're not there. And then over about a 3-minute period, 2-minute period, they pass us by and then they disappear again. So this is sort of a really big problem for biological and chemical oceanography, for that matter, is this issue of patchiness in the ocean, and that our traditional sampling methods just do not, they really don't do, aren't adequate to describe this sort of patchiness. So Brad and I and some others started to scheme on this, and we were able to, Brad effectively was able to get some funding through the Navy. Navy has an interest in this, obviously, because these kinds of aggregations can play havoc and wreak havoc on acoustical and optical instruments that Navy uses for looking for enemy targets. So today I'm going to mainly talk about our field campaign. It does include some big data type analysis. We're doing some machine learning work as well as numerical modeling. All right, so the cruise, we did this a couple of years ago off the coast of New Jersey and Delaware, sampling this grid here. And the idea was we wanted to try to look for these intermediate trophic levels and their association with fronts and eddies. We've gotten very good over the last decade with predicting with models these fronts and eddies, as you can see here in this image from Null School. And so since we can predict those pretty well, if we can understand the association of the intermediate trophic level with these fronts and eddies, then we might be able to predict those as well. So this is sort of what the experiment looked like in its conceptual phase. And I'll note here that we had a lot of what I'll call leading edge technology, and maybe we were bleeding edge for a lot of this, because frankly, when we got out of there, a lot of this stuff ended up not working. It's pretty frustrating. But we did actually get a lot of optical and the optical imagery data that we were able to put together that I'm going to show you today is pretty cool. Our part, Alexis Hagemeyer is one of my grad students, and she's doing her thesis work based on this project. So she and I were on this cruise together. Our work really involved sort of that lower trophic chemical aspect, quantifying nutrients, phytoplankton production, respiration, and microzooplankton grazing. And I'll leave that for Alex, who's hopefully going to defend her thesis this fall. So we have a publication that's pretty close, I think, to being accepted. It's being led by Adam Greer, and it really focuses on a comparison of different gear sampling types and what we were able to see with all of these optical images that I'll show you here in a second. So what we did is there were a number of instruments that we towed across these red and blue lines. And not only do we get all of this rich biological imagery data, but we also have CTDs and other instruments that are strapped on to the towed package. So we're able to see the biological community in the context of all of this very rich physical, chemical, and hydrographic information. So you can see, for example, on this first tow from I-5 to I-12, we passed through a very strong salinity gradient front here as we moved offshore from Delaware Bay influence to more oceanic waters. So, you know, natural history type work, and to me this is really fun. It's really hard to publish descriptive ecology, but we're really close with this paper in terms of just describing this broad marine life spectrum that we observed in relation to biophysical characteristics. So just quickly, here's sort of the optical or the imagery systems that we deployed. We deployed a cytosense, which is a scanning flow cytometer, a holocam, which is a pretty cool instrument that uses a laser, and then it deconvolves the scattering and the backscattering, and you're able to put that together again as an image, a holography image. And then we used this instrument called the ISIS, the In Situ Imaging Ichthyoplankton Imaging System. And they have different characteristics. The cytosense can see things very small down to about a micron in size. The holocam is sort of imaging the next higher size of particles from about 5 micron all the way up to 40 millimeters. The ISIS camera, there's 2 cameras, a small and a large camera, that are imaging in the range from about 42 microns up to pretty large size of about 50 centimeters or so, almost half a meter. So what's cool about this imaging is that we get to see this broad spectrum. And you could do this, well, you really couldn't do it with traditional net sampling. You could collect these phytoplankton and zooplankton, but as you'll see, you wouldn't get the kind of context that we're seeing. But this is just a general overview of what we see across these different space scales and different imaging systems. So things as small as a dinoflagellate, even some very small ciliates, as you can see, surrounding this bigger copepod. And then as we get up to bigger scales, we see pteropods and echinoderms and appendicularians in their houses. This is what Alex Hagemeyer is working on. And then as we get up into even bigger sizes, we see all this rich diversity of gelatinous zooplankton, jellyfish, etc. And so we wanted to, just a little bit more detail. So this is Alex Hagemeyer's work, a little bit more about the ISIS. As I mentioned, it's towed behind the ship at a speed of about 2.5 meters per second, and it's undulating. It's a toyo, so we get different depths, and we can see these organisms vertically. It's a big data analysis. The imagery generates about 400 gigabytes of data per hour. Currently, Alex is doing all of this classification manually. But we are now beginning to work with some computer scientists to do machine learning using these data. But just, I could probably just put this on random play for the rest of the seminar, and I'd be happy just looking at all of these images. But just beautiful images of these gelatinous plankton that ordinarily you would not see this in the real world because net tows destroy these. And so literally we are seeing things that nobody has ever seen before in terms of where these animals are located, how they're co-located with prey items, their orientations, and so forth in the water column. So one of the things we did in this Greerdahl paper was just to simply look at particle size, the number of particle or different particle bins, and their abundance. And you've probably seen figures like this in textbooks before. We all kind of have a general sense of this, that larger animals are less abundant than smaller animals. So, out here at 1 centimeter in length, maybe we only are seeing 100 or so of these individuals per meter cubed. But then when we get down to very small particles, now we're looking at, billions of individuals per meter cubed. And sort of the point of this figure was that all of these imaging systems have the same slope of this fit. So they're capturing this relationship of particle size and abundance. And so, you may not appreciate it, but being able to see these things over such large magnitudes, we're looking over 3 orders of magnitude and scale here. So that's going from something like this, about 1 foot, up to the size of an aircraft. So extremely large scale that we're able to observe with these imaging systems and put it all together in the same location. So one thing that was pretty cool that we sort of made some hay about in this paper were these very thin layers, but very dense aggregations of ciliates that we observed with the holocam. So on the right here is shown a vertical profile. The holocam is a vertically profiling instrument as well. And it's just showing the particle concentration versus depth over 4 different profiles. So on each one of these profiles, we see this spike in the particle concentration. You know, here it's at about 18 meters or so, here at about 23 meters. But what these are extremely high densities of ciliates, and it's the ciliate, the strombidium ciliate that is in these high densities. And with the holocam, we can see the association then of predators. This is a big Calanus thinmarchicus copepod, and it's surrounded by these ciliates. So again, we're seeing things in nature that literally have not been seen before. And don't these just look like bite-sized morsels, you know, for this big guy? This is like the perfect size based on theory for what this guy would be eating. So we also compared net data with the imagery data. So net data is sort of the standard of oceanography for biological oceanography. And just really quickly, what you'll notice here is that the net data tells us a story that basically the copepods are dominating everywhere. We can see that in this first net toe, it's almost all copepods. In the second, it's a bit more of a mixture, but still mainly copepods. However, when we look at the imagery data, the ISIS data that captures all of those gelatinous zooplankton, so I'm going to draw your attention here to sort of this range in here between about 0.3 and 0.7 millimeters. We can see at smaller sizes, yes, the copepods are dominant, but then when we get into this intermediate range, the gelatinous zooplankton, the appendicularians, a lot of these soft-bodied organisms that are typically destroyed by nets are numerically dominant. So to me, this has completely changed the way I view this intermediate trophic level in the ocean, which is really cool when you're surprised by data like this. So I'm just gonna go through this quickly. I ran a little bit long on that section. Sort of the take-home message is that the communities are very patchy. You wouldn't be able to see this with net data. And then something for modeling that we'll think about going forward is that our simple food web, that model that we usually include like a copepod grazer, it's just grossly oversimplistic and not adequate. So something we'll look at going forward is how do we improve representation in that intermediate trophic level. Okay, so bringing it on home now towards the Gulf of Mexico. This next section I've spent most of my career working on hypoxia and the relationship with nutrients and eutrophication in the Gulf of Mexico. Over the last several years, since I've been at South, I've been fortunate with some collaborators. We've done a number of cruises, and we've been looking at the role of coastal sediments and the lower water column in carbon cycling and how it contributes to hypoxia and acidification. These are really important questions. Approximately 20% of all river, 20% of river 80% of all river suspended matter is deposited in the shelf environment. Only 20% makes it off the shelf. So most of the terrestrial carbon cycling is happening in these nearshore sediments. Where there's large river inputs, there's also extreme phytoplankton eutrophication production that can also drive organic carbon respiration and deposition. And we published this now about 10 years ago, some work that was led by Wei-Jun Kai based on some of our earlier cruises, where we link hypoxia and acidification. So I'm just going to walk through this quickly, and then I'll probably speed dash through the rest of the slides to try to get this done within 45 minutes. But effectively, when we look at sort of a river-dominated system, we've got inputs of nutrients, we've got inputs of organic matter, and sort of our classical paradigm for eutrophication is that these nutrients stimulate primary production, creating more organic matter. And this is linked to hypoxia then because that organic matter sinks to the bottom, and that organic matter is then respired. And in that process, it consumes oxygen and then it also creates carbon dioxide. So there's a respiratory part of the acidification, and then there's an open ocean, sort of the climate change part of the acidification. In the open ocean, the ocean's taking up carbon dioxide because of processes like I described with the spring bloom. And then carbon dioxide is a weak acid in water, and it creates a proton that effectively drives the pH downward. So in the work I want to talk about today, we're going to look at actually, the previous work looked at sort of the water column aspects of this, and more recently we've been looking at the biogeochemistry of the sediments and how this is driving further acidification in these waters. So we did a big synthesis of previous cruises as well as the recent cruises that we've done in 2017, 2018, and 2019. Synthesized all of the summer CO2 system chemistry that we've collected from the shelf. So all of these stations shown here on the right. And then also, as I mentioned, in these more recent cruises, we've been measuring the efflux, the flux of material from the sediments, the CO2 system fluxes from the sediments, so we can quantify their contributions. So how do we do this? We collect sediment cores using a multi-core, or in some cases a box core. The sediments are collected into these acrylic core liners, and we can take those into the lab on the ship. And then we can set these up, and over time then we can measure the change in concentration in the overlying water, and that's what's shown in these graphs here. So this is an experiment where we were measuring the change. Well, actually, we measure all of these parameters, but these data show the increase in the dissolved inorganic carbon, and then the commensurate decline in dissolved oxygen. So these are the sediment respiration that's controlling the changes in this chemistry. So this is a little bit of a busy figure. Basically, what we wanted to see is if those sediment fluxes, if the ratio of the alkalinity flux to the DIC flux is less than 1, then we should expect the pH to drop. If the total alkalinity is greater, total alkalinity to DIC is greater than 1, then we have buffering, because the alkalinity can buffer any additional protons, and so the pH doesn't drop in that situation. So what we were looking for is benthic fluxes where the TA to DIC ratio was less than 1. And what we found from our data was that it was actually about 0.5. And so when we look at the relationship between dissolved oxygen and pH, under low oxygen conditions, the low oxygen is created because of respiration, taking up oxygen. And the low pH is also created because carbon dioxide is being created. And so there's a lot of lines on here. The green line is really the important part for our work, because what it shows is that when we have this low TA to DIC flux ratio, and we get down to anoxic conditions in the sediment, which the sediments pretty much are anoxic below about 1 millimeter or so in the surface sediments, but that anoxic biogeochemistry drives the pH down by about 2 units. So this is pretty significant. And I'm just going to skip to this less busy figure, our conceptual model of what's happening. And I'm going to look at the, we just talked about the pH. I'm going to look at the aragonite saturation, which is the omega. So omega, if it's greater than 1, it means that, well, if it's less than 1, it means that calcium carbonate structures are going to begin to dissolve. And so a threshold associated with acidification is this aragonite saturation level. And what we see is that in the year 2100, when we combine the effects of respiration with ocean acidification, plus the benthic biogeochemistry, we are now driving that saturation in those very bottom waters to less than 1, which is going to begin to dissolve calcium carbonate shells and other structures. All right, so this last part with the last few minutes, I just want to discuss what we're doing now, how we're kind of bringing all of this previous work and our understanding and models and deploying these in Mobile Bay. So we were recently funded to begin some new work to characterize the environmental trends and variability in dissolved oxygen, pH, salinity, and temperature in Mobile Bay, and trying to relate those ultimately to higher trophic levels, things like oysters, blue crab, and speckled trout. So we want to understand how is that forcing climate, changing river conditions, changing nutrient loads, how is that affecting oxygen, pH, salinity, and in turn, how is that affecting things that people care about like oysters, crabs, and fish. And so in this work, we're using our model, but we're also going to be adding a numerical coastal food web. And so it's the big project is a very interdisciplinary effort that's been funded by NOAA Restore. It's potentially a decadal project, so we're just thrilled about this project. To me, this is perhaps something I'm most excited about in my career, because it's very rare to have potentially a decade of work like this ahead of us. So I'm going to skip a little bit. So what we're doing is we're employing field methods, experimental methods, and modeling. I'm just going to walk through these very quickly. This is the station grid that we've begun to sample this summer, and we'll continue to do this monthly, hopefully over 10 years, and we'll be able to develop a very good idea of both the spatial and temporal variability in our key state variables, how those are related to primary production and respiration, and in turn to pH, oxygen, and DIC. Along these lines, Ronnie Baker, Sean Powers, and Juice Sebrian are also employing a field program to quantify recruitments, abundance, and community composition that over a decade, hopefully we can put together a time series and a story that relate these environmental trends and patterns to community composition and biomass. Part of the work is experimental. We want to be able to know what thresholds affect higher trophic levels. So we have a good understanding of like individual thresholds. For example, hypoxia is a well-known threshold of oxygen that's 2 milligrams per liter. Below that concentration of 2 milligrams per liter, animals either die or flee. But what happens when we begin to combine other stressors like low pH, high temperature, variable salinity? How does that change that oxygen threshold? How do these combinations in turn affect all of these other thresholds? So we're really excited. I think many of you have heard the news that we're getting a new wet lab building at the Sea Lab. Hopefully it will break ground later this year. And we also have some funding from the Center of Excellence through the BP oil spill settlement. And with the new building and that funding, we're going to be able to build this new experimental system that allows us to do these kinds of multiple stressor experiments. And this will be a resource that's available to everyone at the lab. And then finally, we're bringing this all together through the modeling component. There's a hydrodynamic model component for the bay. There's a watershed model, so looking at predictions and scenarios for the future land use development in the bay. The biogeochemical model is sort of that conceptual model and the numerical model that I've been talking about. We're building with the fisheries folks the food web model, and ultimately we're linking that to ecosystem services so that managers can make decisions. And so one of the emphasis of the Restore, the NOAA Restore program that's funding this work, is that the outputs and products really have to inform management. So we're working very closely with our partners at the National Estuary Program, the Alabama Department of Conservation and Natural Resources, as well as the Nature Conservancy to make sure that what we're producing is useful to them. And with that, I'd just like to thank the funding sources for our work, as well as all of the collaborators. And I'd be happy to take any questions if there are any. Thank you. Right,
Hey, John, this is Jeff Krause. I have a question for you.
Yeah, hey, Jeff.
Regarding the Greenland blocking, how is that atmospheric phenomenon different from the North Atlantic oscillation? Because that's looking at the pressure gradient as well between the Azores and Iceland. And that has actually been explained to, a lot of the patterns they see at Bermuda at Bats with how that oscillates explains a lot of the productivity shifts there.
Yeah. You might have been one of our reviewers because we had that exact same comment.
Wasn't me.
So yeah, we've looked at a number of different climate indices. Did you say the AMO or the NAO?
NAO, North Atlantic Oscillation.
NAO, right. So it is based in Iceland, and we did look at it's sort of similar, but they don't. The NAO doesn't track as well with the movement of the jet stream, and so we sort of, and also the NAO, it doesn't have the same mechanistic interpretation as Greenland blocking. It is a index of geopotential height, but we figured or decided that the Greenland blocking, because it is mapped over that large area, for one, it seemed mechanistically to represent the process better. And then we also looked at correlation between Greenland blocking and NAO, and there is a correlation, but they're not exactly one-to-one. So they are sort of similar, but they do seem to be kind of tracking different atmospheric phenomena.
Hey, John, this is Lee. I had a question for you.
Yeah.
First of all, excellent talk, by the way. I really enjoyed that. I had a question. I like the part about using the camera instead of nets to look at animals without having to destroy them. I think that's really novel and exciting. And I was wondering, you mentioned that you're seeing a lot more gelatinous animals when you use the camera. How do you think that's going to maybe change higher trophic levels and energy flow in those kind of models? If you could elaborate on that just a little bit.
Wow, yeah, that's a hard question, right? So the modeling that I've done thus far has really we've always kind of treated the zooplankton as like a closure term on the model. It's sort of a fudge factor. We can turn that knob if we're getting too much production. It's not very satisfying or mechanistic. So we've really just started to explore how we can, build the higher trophic levels into that food web model. The main thing is that clearly having, the copepod in the model as the dominant grazer is incorrect. So if we start to think about gelatinous zooplankton, I'm not really sure if we know. Well, some things like appendicularians, a lot of things eat those, and that's well known. But when we start to get up into the jellies and into siphonophores, it's not very well understood at all. Not only their life histories are poorly understood, but then their trophic interactions are few and far between. And a lot of that is because they're not easy to collect, and so you can't bring them back to the lab easily and do experiments. And then they're just trying to observe them in nature. And what we've observed so far is that we see, I had an undergraduate student, Hannah Wood, that looked at the pteropods, those veligers. And what we see with the pteropods is that there's a lot of hydrogondusae that tend to be around where we see really high densities of those pteropods. And so we're inferring that there's some sort of a trophic interaction there, but that's sort of the level that we're at now with these data is, just trying to look at who's co-located in space and time, and then make inferences based on size about who's eating who. But beyond that, I don't think that the science is really there to address that question of how that then goes up higher trophic levels, at least that I'm aware of. See, I see a chat
question in the chat. Yeah.
Okay, yeah, there's a question from Kelly Major. She asked, what's the primary composition of the phytoplankton blooms? It depends. in the extreme North Atlantic, you can see things like coccolithophores. In the sort of subtropical, it's, you know, a mixture of probably cyanobacteria is one of the main dominant guys out there during the non-bloom, but then during the bloom itself, I would expect, you know, we're seeing more diatoms. But yeah, I don't really know in the North Atlantic bloom. One of the things that Barenfeld and Boss proposed is that looking at the composition at the end of the bloom is really not that informative because it's kind of the leftover. And so they sort of, in their conceptual model, really play up this idea that we need to have a much better understanding of phytoplankton succession. Who are the guys at the beginning that are triggered by the conditions? They perhaps grow up, but then they probably get grazed down and are replaced by things that are, you know, not as, that aren't as edible. So this is I think an important question, but it's something that more research is needed.
Yeah, I was curious about how things turned over and if you saw any big sort of seasonal changes as the seasons progressed, but you just answered my question. Thanks.
Yeah, so it's interesting actually with the imaging systems. I was really struck by how many big diatoms we saw. I didn't really highlight those, but with the ISIS camera, there were diatom chains, you know, 5 millimeters long. And when we sampled there on the Delaware shelf, it was sort of late April, early May. So it's kind of at the end of the spring bloom. And those big diatoms seem to be what was dominating there. And so, you know, those things aren't easily eaten. You know, they're so big that most of the smaller intermediate trophic levels can't, you just can't handle them. So, you know, again, it's the question of sort of the chicken and the egg. Is like, are those the guys that started the bloom, or are they just what's left over at the end because, you know, only the non-palatable, non-edible stuff is sort of left for both the phytoplankton and probably for the intermediate folks.
All right, I guess. Thank you, John, for your very excellent talk. Yeah, that's almost 1 p.m. Let's see you next week.
All right, thanks everybody. See you later.
Thank you. Bye.